Hello, everyone. So my name is Dr. Nikita Walz, and I think most of you probably won't be too familiar with me. So my camera's not on. There you go. Now you can actually see who I am. So I'm a fairly new member of CFM. I only started last year at the university and joined CFM not too long after. So I'm actually based away from Q Block over in W Block at Toowoomba, and I'm part of the School of Health and Medical Sciences. So I'm actually a lecturer in biomedical science over here. So I'm a biomedical scientist myself who is kind of spread throughout quite a few different research areas in my short research career. So I came over from Western Australia last year, and my area of research and expertise was in cell signaling and basically cell biology. So I was working in my PhD on fertility. So I was working in clinical trials with fertility patients and looking at the cell signaling pathways in reproductive cells. Before that, I was a diabetes researcher. In my postdoc, I moved over to anti-cancer drug development, and now I moved into another whole new area, but my expertise kind of always falls back to cell signaling and cell biology. So bit out of my depths when it comes to the majority of the CFM expertise, but I'm learning lots from you guys. So thank you very much. It's really nice to be part of it. And thank you, Molly, for organising today as well. I really appreciate being invited to speak. All right, so I'm also part of the Simple Hub Med Technologies group as well. So I do a bit of research with Simple Hub. But today I'm going to just present about the biocompatibility of human-derived cells with polyether, ether, ketone, or PEEK. So Molly's already done a lovely acknowledgement of country, so I might skip over that in the interest of time. And this work is actually dedicated to Margaret Ann Vella. So the reason that this project came about is we were approached by a private inventor. He came to us with a device in mind, a bit of a prototype, and he wanted to test this potential medical device. And it came because his wife, Margaret Ann, had a stoma, which is what I'm going to talk about a little bit in a moment. But she had this medical procedure and she had a lot of complications from that. And he wanted to find a way to actually give her some relief for all this and to give that to other people in the future as well. And John's actually an engineer himself. So he came to us, we came up with a bit of a project plan, he was happy with it, and then we have moved forward. So we've got some preliminary results here, but it's still very early stage research and going to be very different to what we just heard about from Min. All right, so before I get started, I have a couple of graphic images in the next couple slides. So I do want to give everyone a little bit of a warning. There is some sensitive content. So if you do not do well with any images of bodily or anything graphic, please make sure that you turn away over the next few slides. So what is a stoma? A stoma is basically a surgical procedure where you actually take part of generally the bowel or part of the urinary system and you actually externalize it to the abdominal wall. So this is also known as ostomy. And it involves that exteriorizing portion to the outer surface so that you can actually extract either urine or fecal matter from that stoma. So it's generally implicated for people who have inflammatory bowel disease, colorectal cancer, diverticulitis, even abdominal traumas as well. And in Australia, we have about 46,000 people currently living with a stoma, and worldwide there's numbers increasing as the years go on. So we're starting to see a lot more bowel cancer emerging and stomas are becoming more and more common. And there's typically three main types of stoma. There's a bunch of subdivisions as well, but the three main types are colostomy, ileostomy, which are two types of bowel stomas. So they just come from different parts of the large the large bowel. And then we've also got a urostomy, which comes from a part of the urinary system. So these can either be temporary solutions or they can actually be permanent and people have to live with these for the rest of their lives. So I'm just going to show what a stoma looks like on the bowel, the bowel coming out of the abdominal wall. So if you do want to look away, now would be the time. So it looks like this. So you can see it's actually kind of attached to the outside wall, so they have to stitch it in place. And generally it's covered with a colostomy bag like this one here to actually catch that fecal matter or urinary matter, depending on what type of stoma. So the problem with stoma is that it comes with a lot of complications. So it's a good solution for a lot of conditions, but it does have a lot of side effects that come with it. So we've got them kind of divided into early stages, which happen within 1 month, and we have some late stage complications, which are generally after 1 month. So some of the early problems we see, which is what our inventor came to us with his wife, she had a lot of skin irritation and she had it very long term. So this skin irritation can lead to dermatitis. It's basically breaking the skin barrier. Our barrier of our skin is one of our innate immune parts of our body that help protect us. So if you break that, you're more likely to get infection. So these people have a very high incidence of infection in the peristomal area. You also have the risk of stomal necrosis, so that tissue dying off, bowel obstruction as well. So sometimes the stoma can actually change its size. It can reduce in size, and these people need to have them dilated. And also things like diarrhea, stoma retraction. So that's where the stoma actually goes back into the interior portion of the abdomen. And some of the late stages include stoma stenosis, prolapse, peristomal hernias, fistulas, and a whole bunch of other complications. So I have a couple of images that are going to show you what this looks like. So again, if you do not do well with these images, please look away. But here are some examples of this skin irritation and infection at the site of the stoma. So down the bottom here, we have some examples of, for example, stoma retraction, where it's actually moved back in. So that would require another surgical procedure to correct. We have some examples of some necrotic tissue starting, some infected tissue, and also some stenosis as well. So these are some common complications. And one of the awful things is once they get this irritation, they have to put a colostomy bag, which has a sticky adhesive on top of the skin that's already irritated, broken, potentially infected, and it just repeats the cycle and makes it worse and worse. So these people really, really struggle. So there's such a need for a better solution for these individuals. So what do we know about stoma and quality of life? Well, there's a study that was conducted in 2016 to 2017, which is the Stoma Quality of Life study. And they looked at some of the sort of parameters around stomas, who was suffering them, what their quality of life looked like. So from their study, they looked at 45% female and 55% male users. So it's pretty even across genders, but it can affect anyone essentially of any age. So we have people from 18 to 29 years old, all the way up to 70 years old. So the most common group is actually 60 to 69 and that later older years. So it is more common in elderly, but it is also starting to happen earlier as well with the incident of bowel cancer. About 80% of these people were living with a partner, so they had some at-home support, but there is a big discrepancy in the people who are living rural versus the people who live in a metropolitan area. So we know that people who live rural or regional have less access to healthcare, especially if they're very rural. We have people in metro areas that can regularly have a stoma nurse come, change bandages, check skin irritation, put creams and all that sort of stuff and have interventions, whereas people living rural, regional do not have that access or that ease of access. So there's a big issue for those people. And a couple of other things that are related to quality of life, but not necessarily the complications that I just mentioned. About 39% of people reported being worried about leakage. So that's another thing. If you're having leakage of urine or fecal matter to that area, it causes, again, skin irritation, dermatitis, infection, etc. So it makes that whole cycle repeat. And about 44% said that they experienced their ostotomy pliants visible during clothing. So it also makes these people feel very insecure, and it can be quite mentally damaging to these people. So there's lots and lots of research in the quality of life and that there needs to be an improvement for these people. So one of the ways that we could potentially look at improving the life for these people, this quality of life, is through alternative medical implants. So at the moment, it's basically a stoma. There's not much else. It's just a stoma. But we are investigating PEEK material. So this polyether ether ketone material. It's a semi-crystalline thermoplastic polymer. It has a very, very good resistance to temperature, up to about 300 degrees Celsius. And that's great for medical devices because obviously we need to sterilize medical devices. So we want to make sure that it withstands sterilization procedures. It also contains a main chain of a macromolecule composed of benzene rings, a ketone, and an ether group. One of the other positives about it is that it's non-magnetic. So compared to things like titanium and other things like that, it's got reduced thermal conductivity. So one of the problems if you are putting in medical implants is that they can show up on screens. So if you're doing an MRI, CAT scan screening, it can actually occlude other material behind. So if you're doing a screening in that area, you might miss something, it's blocking it. And of course, being less thermal conductivity is great if you are doing something that requires temperature. So PEEK and its composites do not elicit systemic toxicities that has been shown previously in lots of studies, or adverse local tissue damage when implanted. However, those studies are limited to dental and orthopedic implants. So there's a few examples over here. You've got the chemical structure of PEEK and some of the temporary implants. So there's quite a few different types. We've got almost like screw types for dermal implants, some sort of plug pegs type ones. So there's a few different variations of that, but again, it's only in real bone tissue. So there's no real investigation in soft tissue, which is what we wanted to look at. So before we get started, we have to make sure that all the research we're doing falls within the international standards. So we are using the ISO 10993 guidelines, which outlines the biological evaluation of medical devices. So I've had a really fun time spending lots of time reading many, many pages of the ISO guidelines to make sure that our research falls within these guidelines. So just to give everyone a bit of context, the implanted devices are subdivided into 3 classes. So we have devices that are designed for limited exposure, so less than 24 hours, prolonged exposure, which is greater than 24 hours but less than a month, and permanent devices, which are greater than 30 days. So we are obviously wanting something more permanent. So we're looking really at, see here, the permanent devices. Because our device will be something that's going to be in the anterior abdominal wall, which will sit at the surface level, we have, again, surface devices subdivided into 3 categories. So we have intact skin devices, mucosal membranes. So a mucosal membrane would be something like the oral cavity. And then we also have breached or compromised surfaces. So ours is sort of an intact skin device, and obviously there's a risk of breached, compromised surface. So we have to kind of fall between both of these. And several studies need to be undertaken in order for these plastics to be FDA approved. So one of the things that we have to look at is cytotoxicity studies. So we know that systemically there's no toxicity with PEEK. We've shown that in previous studies. So we don't need to look at systemic toxicity, but we do need to look at the localized cytotoxicity. So a lot of our studies revolves around that. We also need to look at immunotoxicology, so specifically in the dermal environment and in the stomal cells, we need to look if there's any immune interaction with PEEK. Does PEEK elicit some sort of immune response where it wants to attack the tissue because it's being exposed to PEEK or something like that? Some other things like dermal sensitization, they actually come later down the track. So we're really looking from a preclinical point of view, whereas later down the track in clinical studies, then you would start to look at things like dermal sensitization and some of those other ISO guideline requirements. So our studies really focus on cytotoxicity and immunotoxicology for the early stages. All right, so what do we know so far about skin biocompatibility and PEEK? Basically very, very little. So in the context of skin as an implantable device, PEEK has only been assessed in 1 study, and it was an ex vivo study. So it's never been done in vivo or in vitro even. So basically this study created a bit of a cylinder-shaped device with PEEK. They compared it to titanium implants in an ex vivo porcine model, which is pig skin. They cultivated this in nutrient-rich media for 2 weeks, and then they did some biomechanical testing and some histomorphometric studies as well. So from this, the implants were then processed for the degree of epidermal growth. So that's the growth of the upper layer of the skin, and also the tissue implant contact. So how these actually interact with one another. And from this study, they showed that PEEK implants had better tissue integration with soft tissue compared to the titanium implants. So that was very promising, but of course, this is not a very good representative model of what's happening in our bodies in the case of implanting a device. And this is basically showing the layers of skin down here of where that would sit. So it's kind of embedded in this top layer of skin and the middle layer, the dermis. So you've got the epidermis at the top, the dermis, and then the hypodermis is where your subcutaneous fat cells sit. So it's embedded in the skin like so. Okay, so where does our project come into it? So our project we've titled the Velostoma Freedom Project, and the overall aim of our project is to essentially assess the cellular interactions of various human cells, specifically ones that would be implicated in a stoma. So that's going to be the skin cells, so keratinocytes, our colorectal cells, so colon cells from the bowel, and urothelial cells, which are basically the cells that line the inside of the urinary system. So we're going to expose these to peak material to investigate the biological responses related to some of those cytotoxic and immune studies. So specifically looking at cell proliferation. So do the cells actually grow on that surface? Do they survive and are they happy on that surface, i.e. are they viable? Do they adhere to that surface and stay on that surface? Do they migrate to that surface? And also then we're going to look at if they elicit an immune response in a negative way or even a positive way. So this is essentially what the prototype looks like down here in figure 1. So this is basically a flange that's going to sit under the skin surface, so around the fat sort of layer, and then underneath the fat, underneath the fat layer there is muscle. So it's going to sit basically underneath the fat layer on top of muscle. So there will be some changes probably to the device over time. So this is a very early prototype essentially. But this is actually created by injection molding. So our inventor got this injection molded and gave it to us like so. And actually on this surface here, so this is the surface that would be facing sort of underneath the dermal layer here, so facing upwards to the dermal layer. So these bits are actually smooth. So we refer to this as an unmodified surface or a smooth surface throughout our research. And then the outer layer here, or the outer ring, and also the back of the device, so all of these portions are all a rough modified surface. So we're calling it rough because as you feel it, it's very, very obvious that it's rough. It's got some sort of micro grooves on it, so the surface topography is quite different on each side. And that was intended to basically help cellular adherence. So many studies show that the more modified or rougher the surface, the better cell adhesion is. So it actually helps promote adhesion. And then John has also invented a bit of a cap that the idea would be that this sits under the skin. This bit would be open up, so the stoma would actually be stitched to the inside. So in this case, this is a very small version, so this would be for a urostoma. So it would actually be stitched to the inside here. This bit would be open, and then you could actually attach a bag like so to the outside, and you could always cap it off if you want to go for a swim or something like that. So these patients, because they have an open wound essentially, they can't just go for a swim or bathe the same way that anyone else can. So hopefully we can create some sort of cap. I don't know if this will be 3D printed as well. We haven't really thought about that too much yet, but that would go on top, be sealed tight so that we can close it off when necessary. So that is our first initial prototype that we're working with. And what we do is we typically just stamp out pieces of the device and we compare the smooth and the rough surfaces. So we have a little bit of a steering committee going for this project. So we basically wanted to establish a bit of a committee to give us guidance because we're working over so many different areas here. So we've got some insight from clinical nursing. So Dr. Sharon Rees, who works over with me in W Block, she's an academic, but she's also a very experienced stoma nurse. So she's coming from the clinical nursing point of view of the sort of application of a device like this. We've then got our surgical consultants. So we have Dr. Tom Arthur and Dr. Johnny, who's I'm sure most of you have probably met or seen around CFM. We then have Molly, who's a bit of a consultant for the research side of things. And obviously she's got expertise in peak, which is why she got thrown in there as well. So we appreciate having her on board in whatever capacity. So Dr. Anna Krusik is one of my fellow lecturers here in Biomed. So she's also working on the project as a biomedical scientist along with myself. And we also have Professor Eliza Whiteside, who's basically the head of the steering committee, and also our Simple Hub co-directors, Polly and Triss, are also involved. We've also got a consumer. So Zoe is someone who actually has a stoma. She's very involved in the community in terms of stoma support groups and things like that. So she comes with her point of view, what she thinks would work for a consumer, and we kind of adapt and test things based on that. And then we, of course, have our inventor, John Vella, and also his granddaughter, Mia, is also very involved. And she's actually studying a science degree and does a lot of 3D printing herself. So she's very interested in the project. So that is kind of everyone involved. So I'm just going to do a little bit about the preliminary data so far. So the first thing that we wanted to look at when we got the device is that we could physically feel that it had a different surface topography on each side, but we actually wanted to test that. So we characterized the surface topography using a surface roughness tester over in ZBlock. So we looked at the smooth surface over here compared to the rough surface, and the modified or the rough surface was significantly rougher, which is what we anticipated, and the RA value of 4.93 compared to the unmodified or this smooth surface, which was 0.59. So it was significantly different, and we've just worked it out as a ratio there to show the difference. And then we actually looked at that under SEM as well, so we could actually visualize that difference. So over on the left here in figure 2, you can see the smooth surface. There's not much to really look at compared to the modified or the rough surface on the right hand side. You can see some of those grooves and those the sort of elevations of the device. So the next thing we wanted to look at, so we kind of started looking at the surface before we really put cells with it to make sure the surface was compatible. So another thing that we wanted to look at was our surface water contact angle. So essentially, we wanted to see how hydrophobic or hydrophilic the surface of the device was, because that's obviously really important when we start to add cells and cell culture media and things like that to the material. So we wanted to make sure that the cells would actually like that environment. So typically, if a water contact angle is less than 90 degrees, the solid surface is considered hydrophilic. And if the water contact angle is above 90, it's considered hydrophobic. So we wanted it to be smaller. So we wanted a low contact angle. So we took multiple samples for each segment type and used 3 distinct points to test water contact angle. Very, very simple. Just put a drop of 1 microliter of water pipetted onto the surface and then measure the water contact angle using the following equation over here. And we looked at multiple time points. So one of the fun things when we were looking in the literature was that there was no consistency across the board of water contact angle in terms of when it was measured. So we just measured it over several time points that was found in the literature. So we measured the image after 10 seconds, but we actually looked at 3, 10, 30, and 120 overall to see what point that water contact angle changed. So what we found is in the rough surface over here that we saw a decrease in the water contact over time. So at 3, 10, 30, and 120 seconds, there's a big drop in water contact angle. And you can see it in the images here. So the first image on the left-hand side is at 3 seconds. So you can see that there's a nice round droplet of water. And then you can see that contact angle changes significantly at 120 seconds. Whereas in the smooth device, there's not too much change across the board at 3, 10, 30, and 120 seconds. So it basically, the water contact angle stays quite consistent. So from that, we knew that the rough surface had a better environment. So it was more hydrophilic. So the next thing we wanted to look at with our device was a bit of the flexibility. So one of the things that we talked about in our last steering committee meeting, so Zoe, our consumer, expressed some concerns around the flexibility of the device. So we did a quick test of flexibility. So this is only one-time test. We have to repeat this a few times and do some more analysis. But we wanted to use the MTS Alliance RT10 over at P11 with the crosshead attachment to actually measure a little bit of flexibility of the device. So when we applied force, the device actually snapped off at about 322 newtons at 105 seconds, and we applied force every 2 millimeters of force per minute. So you can see there as the flexibility increases with the load and then drops off at 105 seconds. So we have to do a little bit of analysis to adjust some of the parameters here. So this is very, very preliminary results. So essentially, we know that the device has quite a lot of ability to resist the flexibility and the compression forces, and that's what we want to see, because obviously if it's in the abdominal wall, you're going to have a lot of compression when you are leaning over, moving around, if you get hit in the wall, all of those sorts of things. So we wanted to make sure that that device does have enough flexibility, but is still very strong, and it is very, very strong. So the next thing, after we've looked at a little bit of the material testing side of things, we wanted to, of course, start culturing with cells and have a look at what was happening in the presence of the peak device with cells. So all of our preliminary results here are with our human keratinocytes or our skin cells, and then we have to repeat a lot of this work with the other cell types that we're looking at. So the first thing that we wanted to look at was cell adherence. So do these cells actually attach to the device? Because there's no point doing anything else if they don't. So we did cell culture for 24, 48, and 72 hours. We then fixed the cells in a 2.5 glutaraldehyde solution. We did a series of stepwise washing steps with ethanol at various concentrations. So starting at 50, working up to 100% ethanol to wash away the fixation solution. We then did a chemical drying step with a HMDS solution. So we dried them out, left them overnight to evaporate off, and then we mounted our samples on our conductive double-sided carbon tape and got them ready for SEM. And they were coated with a gold sputtering prior to SEM. So this is just a nice SEM photo. This is not the SEM that we have. I use the SEM we have in Z-Block. So Molly has told me that that's what we had available. So we actually did get some nice images. I know Molly wasn't so successful when she tried it, but we actually did get some nice images. So we basically put the device in media with no cells for 72 hours. So it was in the same cell culture medium and conditions. And then we measured the smooth surface versus the rough surface in SEM. So you can again see that the smooth surface and the rough surface are pretty different. So the smooth surface doesn't really have too much indentations or elevations, whereas the rough surface definitely has a lot of micro grooves. And then in the presence of the cells and the device at 72 hours, you can see in the smooth surface, there's not too much. You can see a little bit of bubbling. I think this is just an artifact from the chemical process. I'm not too sure. But you can definitely see where the cells attach on the rough surface. So there's a nice example here. You've got the nucleus of the cell, some of the projections of the cell. So when the cells attach, they have projections that come off and actually will attach with cell adhesion molecules to the surface. So we found that they do attach by 72 hours to the rough surface, but not the smooth surface. So that's pretty in line with what we find in the literature, that around 72 hours is when the cell adhesion molecules are actually expressed by cells, and they actually will adhere to these polymers. Next up, we wanted to look at whether or not the cells would actually grow. So we know they're attaching, but will they actually grow on that surface? And we look at that through looking at cell proliferation. So to do that, we used a very simple stain, crystal violet. So crystal violet is this bright purple solution which stains proteins and DNA typically. So we wanted to put that in the presence of the device. So you can see little pieces of peak here in the wells. We've got 24, 48, and 72 hours. We looked across the board, compared the rough surface and the smooth surface to see whether or not they were growing. And we compared these to control wells, which had just the normal well plate. So the well plate, the cells grow very nicely on. So we just accounted for the same surface area as the devices as a little bit of a control. And then we looked across the board. So at 72 hours, the crystal violet staining for the rough surface compared to control was significantly higher. And you can see that in the staining here as well, that this purple is much brighter. So that indicates the presence of more cells, more DNA, more proteins for crystal violet to stain. So you can also use crystal violet to look at whether or not the cells are not present or they're not adhering, because there will be an absence of the crystal violet dye, because basically if they don't actually attach and adhere, or they die off and come off, they get removed in washing steps. So you'll have a much lighter color like you see in the smooth. So they're not attaching to the smooth, as we know from SEM. So essentially the rough surface is very good for proliferation, and actually is better than the well plate at 72 hours. So they actually encourages proliferation on that surface. So the next up, we wanted to know, now that we know that they're adhering nicely, they're proliferating there, over time, are they actually viable and happy? So they're not dying essentially. So we took the human skin cells and we seeded and cultured them again for 24, 48, and 72 hours in the presence or absence of the peak material. And the control cells, again, were just the standard cell culture plates to ensure the viability was not influenced by having the peak material there at all. And cell viability was not impaired in the presence or absence of, in the presence of the rough or the smooth surfaced material. So you can see over here at 72 hours, it's pretty comparable across the board. So essentially the cells were nice and happy. They were able to metabolize the reagent we use, which is Alamar blue. So basically, as it gets metabolized, it fluoresces, it becomes this pink, bright pink color. So it's usually blue, as the name indicates, and then it changes to either sort of a purpley or a pink color, depending on the fluorescence. And then you just measure that in a standard fluorescent plate reader. Okay, so the next step, which we haven't quite got to yet, so we are going to now look at dermal immunotoxicology. So to do that, we are looking at quite a extensive cytokine panel. So cytokines are going to be sort of immune proteins that are associated with immune response. So you can look at anti-inflammatory cytokines, you can look at pro-inflammatories, so ones that are going to encourage inflammation or ones that are there to counteract inflammation. So we're going to look at some of those, look at the ratios of those and see if the presence of PEEK is causing some sort of immunotoxicology response. So to do that, we are going to put human-derived skin cells in the presence of some immune cells. So the ones that we're using are monocytes. So these are sort of immature macrophages. We differentiate them into macrophage cells and then co-culture them with our skin cells to look at whether or not a pro and anti-inflammatory response occurs or no response occurs. And we have a pretty cool machine to do that called the Luminex 200. It's a multiplex machine, which basically works on simple antibody Luminex bead, so it captures the antibodies and you can basically look at all of these panel in one well of cells. So it's a very, very nice way to utilize a lot of your cell culture supernatant. So we have that over in the PC2 lab in D block. So we're going to look at all of these, and the ones that I've highlighted in yellow here are the ones that the ISO guidelines recommend you look at, and they are all covered in this 25-plex panel. So we have that panel now up in the D block lab, so we're going to set up this experiment very, very shortly. So the future directions and significance of this project. So these are all very important early preclinical considerations when we're looking at the ISO guidelines to indicate potential clinical applications of PEEK, specifically as a dermal implant or even in soft tissue, because it's never been shown before. So we do have to repeat these experiments with other cell types. So we're waiting on our human bowel and our urothelial cells. So that's been the biggest setback for us, is because of the cell types that we want not being available in Australia and not having import licenses. We have to go through a big roundabout way of getting them through material transfer agreements. And if you've ever had to put through contracts to the university, it's a very busy department, so it takes a bit of time. So we've just got them approved not too long ago, and they're being delivered, I believe, in a week's time. So we're very shortly going to get our other cell types, and we can repeat all of these experiments again with our new cell types and compare to skin cells. So this could provide a very important novel implant for people or individuals living with a stoma who are significantly impacted daily by things like skin irritation, stomal complications, trauma, etc., at that peristomal skin site. And we really, really hope that something like this will improve the long-term quality of life for these individuals so that they can have an implant that's there, something they can close off, something that's not going to leak material, irritate the skin, etc. So it's a really, really exciting project, I think, and we're getting some nice preliminary results to work with. So very, very exciting stuff. So I do just want to quickly do my acknowledgements as well, because as you all know, in research, it takes a whole bunch of people to make this happen. So my co-investigators, Eliza and Anna, our inventor, John, our students, Aaron and Francis, they're our honours students currently, which are helping out on the project, our surgical consultants from St Andrews, and also, of course, everyone from CFM and Simple Hub who have been involved, and also the technical support as well, who have helped train us on things over in P11 when we have no clue about anything that's over there, and also the technical support at D Block Labs as well that help make sure that everything runs smooth there. And thank you all for listening. I hope that I haven't bored you with biology, and I hope that if you have any questions, I can answer them and help.